So hi everyone, so for the next video um, we will have to make an edible animal cell model. So let's do this. So this is the materials that we have to use for animal cell. This is the cell membrane. This is the cytoplasm. Cytoplasm is the gelatinous liquid that fills the inside of a cell. It is composed of water, salt, and various organic molecules. Next, we have the Golgi apparatus. Golgi apparatus is an organelle found in most eukaryotic cells. Next, we have the nucleus. Nucleus represents as the max Kindi. Nucleus is a membrane-bound organelle that contains genetic materials of eukaryotic organisms. Next, we have the vacuole. Vacuole is a membrane-bound cell organelle. It is generally small and helps separate waste products. Next, we have the mitochondria. Mitochondria are membrane-bound cell organelles that generate most of the chemical energy needed to provide the cell's biochemical reaction. Next, we have the lysosomes. Lysosomes is a membrane-bound cell organelle that contains digestive enzymes. Lysosomes are involved with various cell processes. Next, we have the ribosomes. Ribosomes are micromolecular machines found within all living cells that performs biological protein synthesis. So this is now the output of animal cell model. So this is now the output of an edible animal cell model. So that's all for today's video. Thank you for watching and God bless.